Hello and welcome to the webinar, Prebiotic Powerhouses, How Pulses Benefit the Gut Microbiome. We'll be exploring how pulses support gut health and the broader functional benefits they may offer for overall health. I'm Milton Stokes and I'll serve as moderator. Today's webinar is sponsored by USA Pulses. USA Pulses is an innovative group of organizations bringing people together to deliver high quality, nutrient-dense, regenerative, and affordable foods. Through advocacy, research funding initiatives, and policy engagement, USA Pulses drives awareness about the health benefits, sustainability, and the versatility of pulses. Today's learning objectives are to define the fundamental principles, structure, and function of the gut microbiome, recognize chronic health conditions that may be affected by the health of the gut microbiome, and describe the role of pulses to improve client gut health outcomes. We have some housekeeping items for you today. All attendees of the live event, you'll receive an email within about a week with the CPE certificate of attendance. You'll also receive the slides and a recording of this webinar. And now for our time today, we love engagement and we invite you to submit your questions using the Q&A function in Zoom. Next, I'm going to provide a little bit of background to help tee up the webinar just ahead of our featured speaker. Dietitians have unique insight into consumer habits, and this survey of more than 800 dietitians, it shows that RDs expect gut health to be one of the top 3 consumer purchase drivers in 2026. Along with affordability and value, gut health is expected to be top of mind, while comfort and emotional well-being, those are also shaping food choices. And that makes pulses a strong fit, whether we're talking about canned or dry lentils, beans, and peas. Pulses are among the most nutritious and affordable foods available. They also sit at the intersection of what consumers are looking for, and that's foods that are practical, comforting, functional, and backed by science. The survey also highlighted the recommendations dietitians are making to consumers, with increasing fiber intake at the top of the list. Pulses deliver about 6 to 8 grams of fiber per half cup serving, and that makes them an easy, practical recommendation. For most consumers, adding just half a cup of pulses feels realistic and doable. So let's make sure we're on the same page when we talk about pulses. Chickpeas, also known as garbanzo beans, along with lentils, dry peas, and dry beans, these are the primary US grown pulses. And pulses are the nutrient-dense edible seeds of legume plants, and they're often harvested dry and ready for cooking. In addition to fiber, which we mentioned a moment ago, they provide plant protein and other important nutrients. And of course, they're also available in convenient forms, including canned pulses that are ready to eat, as well as dehydrated, pre-cooked varieties that help save on preparation time. A half cup of pulses daily is a practical recommendation for many clients and patients. And one helpful way that you might frame it is as a swap, replacing something people are already eating with pulses. And research supports the health benefits of this small daily change. For example, substituting just a half cup of refined grains with a half cup of pulses each day can help increase fiber intake while also boosting magnesium, copper, and potassium by about 10%. In this webinar, we'll cover the role of pulses in supporting the microbiome and why that matters for healthy aging and chronic disease prevention and management. And we'll look into how pulses may serve as a protective dietary factor in conditions such as neurodegenerative disease, ischemic heart disease, and diabetes. And these are major global health concerns. More than 50 million people worldwide are living with dementia. And ischemic heart disease is projected to remain the leading cause of cardiovascular death up to 2050. And diabetes continues to affect millions of people, with rates rising. In just a moment, our featured speaker, he's going to cover research showing that eating more pulses along with whole grains and nuts while consuming less added sugar, red meat, and processed meats that's associated with meaningful improvements in overall health and longevity. So let me tell you about our speaker. Dr. Ravinder Nagpal directs the Gut Biome Lab and serves as the Betty M. Watts Endowed Professor at Florida State University. His research focuses on the gut microbiome across the lifespan, including its role in early development, aging, metabolism, and neurocognitive health. Ravi's authored more than 150 peer-reviewed publications and is widely recognized through his editorial leadership, professional service, and invited lectures around the world. And so with that, Ravi, I'm going to turn the program over to you.
Thank you, Dr. Stokes, for the wonderful introduction, not just about me, but also about the topic in general. And thank you, to Tracy, to Caitlin, to all the team members for organizing this very timely and important topic webinar. And it's my pleasure and honor to be here and talking a little bit about the research that we are doing in our lab about the nutrition and microbiome and the role of pulses in the gut health. But before I start talking about the research, let me provide an introduction of like why we are doing this research, why it makes sense in terms of health and well-being. Uh, by training, I'm a microbiologist, so I personally believe that we are more microbial than human, and I'm not just making it up. There are data that can prove it by facts. For example, at any point of time in our lifespan, we carry about 100 trillion microbes on or inside our body. And these microbes, they literally outnumber our own human cells by at least 1.3 times, if not more, including all the cells that we are composed of as human beings, like including skin cells, pancreatic cells, brain cells, kidney cells, blood cells, immune cells. If you combine all those cells, our microbes cells still outnumber these cells. And if you compare the genome of all these microbes that we carry on our body and compare their genome with the human genome, then these microbes outdo our genome at least by 100-fold. Again, proving that we are actually basically a living and moving ecosystem of microbes, dominating our own genome, dominating our own eukaryotic human cell organ. So far, we have discovered about 1,200 species of microorganisms associated, fundamentally with human body in one or the other way. But that's only the tip of the iceberg. There are still many, many more bacteria, fungi, and viruses or eukaryotes that have not been discovered yet or understood yet. So probably it might be way more than these species. And we carry a lot of biomass of microbes on our body. Like every time we stand on the weighing scale and measure our body weight, you can imagine like about 1.5 to 2 kilograms of our body weight is represented just by the bacterial or the microbial biomass that is present on our body. And the fun fact is, just like our DNA fingerprint, which is unique from person to person, our microbiome is also very unique from person to person. It varies greatly within individuals. It also, within a given person, it also keeps on fluctuating depending on our lifestyle, our travel, our age, lifespan, relocations, and other factors that are exposomes to us. About all the of all the microbes that we carry on our body, about 95% of that resides in our gut. We call it gut microbiota, gut microbiome, or gut microflora. And that's what our lab is trying to focus on right now. What is the role of this dense, complex community of gut microbes in our health? Centuries ago, the great Hippocrates postulated that all diseases begin in the gut. At that time, he didn't have those kind of technical tools and fancy lab equipments to do research, but he had the wisdom and speculations that something is important in the gut that may play a role in our predispositions to a number of chronic diseases. But look what, after centuries and centuries of advancements of technical tools, scientific progressions, we have come down to the same conclusion. 90% of human disorders have been found to be connected with, directly or indirectly, with something not right in the microbiome. And not just gut issues, not just digestive problems, but even the cardiovascular disorders, specific cancers, muscle and bone issues, respiratory and lung disorders, and even neuroinflammatory and neurocognitive disorders of the brain have been found to have some connections, something not right in the gut microbiome. So that's what we are trying to do right now. One of the focus of our lab is the role of the microbiome in gut health, and trying to discover nutritional interventions that can promote a healthier microbiome. And we are also trying to understand the role of the microbiome and diet in a better regulation of gut-brain axis. And as I mentioned, we carry a lot of microbes on our body, and we are not just, so that also makes sense that we are not just what we eat, but also what our gut bugs eat. They're not just present in our body for no reason. Humans and microbes have evolved together over, centuries and generations. And then we have come down to a moment now that we both cannot live without each other. Many of the human microbes cannot survive when they're outside the human body. And we also depend heavily on these microbes for a number of functions. And as I mentioned, our microbiome is highly unique from person to person because there are so many factors that can decide, that can shape and regulate the microbiome that we carry. As soon as we are born, the way we are born, the way we enter into this world can also decide what kind of microbes we carry. We're going to carry for the rest of our lifespan. Our research has shown in our lab that babies that are born by C-section or preterm birth have a unique microbiome compared to full-term vaginally delivered babies. And then babies that are exposed to exclusive breastfeeding versus formula feeding, they also carry different kind of microbiomes, genetics, environmental factors, demographic factors, siblings, hygiene status, exposure to pets, kids being raised in a farm or in an urban setting. All these factors and our exposures to medications, antibiotics, and other travel and other factors, climatic factors, can also shape the microbiome that we carry for the rest of our lifespan. But of all these factors that decide what microbiome we're going to carry at any point of time, diet remains one of the most fundamental, most profound factor that strongly regulates and shapes the microbiome that we carry at any point of time. Diet has a tendency to change the microbiome very quickly and reproducibly. And these microbes not just predate, on our diet, because every food that we consume is also consumed by the bacteria in the gut because they also need energy from the food to survive and proliferate. But it's not just one-way benefit that they're getting from us. Humans and microbes share a symbiotic relationship where both parties benefit each other. So while we provide these gut microbes, shelter and diet and free food, to survive and make a colony. In return, they also provide us fundamental benefits. For example, this graph on the right side shows that, of course, microbes, they facilitate our digestion. They promote digestions, by facilitating the absorption of a number of nutrients. For example, there are many complex carbs, starches, fibers that human body cannot digest otherwise. So we depend on these microbes to process and digest and metabolize and ferment these fibers. By doing this, these microbes also produces a wide array of compounds and metabolites such as short chain fatty acids that further help our gut functions properly because many of these metabolites act as source of energy for our colon cells. Many of these metabolites also regulate our enteric epithelial barrier or gut barrier permeability. So they also foster our overall colon health and bowel health. By colonizing our gut, these microbes also protect us from invading pathogens because on a daily basis we are exposed to a number of dirty stuff, germs, bugs. Every now and then these things may enter in our gut, but because we have a dense community of microorganisms in the gut, so we don't get sick because these invading pathogens, they don't have any sites. They have to encounter a strong colonization resistance from the indigenous microbiome. So by doing this, these microbes protect us from invading pathogens on a daily basis. And also, they also regulate tightly our enteric immune system and enteric nervous system. So by producing these compounds, some of those compounds also regulate the proliferation of um, neurotransmitters and hormones that regulate our satiety and appetite, such as GLP-1, PYY, GABA, and serotonin, dopamine. So by producing all these compounds out of, fiber degradations and nutrient digestion. These microbes provide us a number of benefits. They regulate our gut health, they regulate our colonic health, they regulate our immune health, they also regulate our gut-brain axis by regulating neurotransmitters and hormones. So we get a number of benefits in return from this microbial community. As Dr. Stokes mentioned, that diet has long been associated with healthy aging as well as good gut health. However, research is only beginning to emerge whether and how microbiome could be playing a role in that link. We know there are specific diet factors that have been associated with good gut health and healthy aging. On the other side, like many studies have shown that poor quality diets that are poor in nutrients, but they are sugar rich or calorie rich, these diets are known to drive inflammations, promote metabolic dysfunctions, increase the chances of chronic diseases associated with aging, and also have been associated with cognitive decline. And emerging research shows that all of these things might be rooted in the gut. On the other hand, a prudent, plant-forward, whole food-based diet, nutrient-rich, not calorie-rich diet, have been associated with good homeostatic and eubiotic microbiome, and this has been associated with healthy aging. So again, emerging research has started to show that microbiome is one of the missing link. On one hand, it can actually uh, be dysregulated because of a bad diet that can lead to increased predispositions to chronic diseases associated with aging. On the other side, it can also be fostered by prudent diet, and then that can provide us protections that can prevent a risk of chronic diseases as we tend to go through aging. Although a healthy diet has always been the priority for all dietary guidelines, not just in the United States, but throughout the world, but recently there has been specific emphasis given to also gut health and the microbiome. This is a pyramid of most recent dietary guidelines in the United States, where there is more than ever, emphasis on the importance of high fiber foods, whole foods. Why these are important for health? Because one reason is these foods are known to promote a healthy microbiome, and that healthy microbiome promotes healthy colon health, healthy gut health, immune health, brain health, thereby preventing the risk of chronic diseases. So we are in a very high time in this where the the role of gut microbiome and gut health is and nutrition in promoting gut health and prevention of chronic disease is being appreciated more than ever before. Our lab is also doing a lot of research on aging, and now accumulating research shows that our microbiome can also age with us. Our research from our lab and other labs have shown that as we move from one stage of our lifespan to another stages, our microbiome also grows with us. We have a unique microbiome when we are in infants, then during childhood it undergoes some other changes. We have a robust and stable microbiome during adulthood, but as we tend towards senescence or aging, towards old age, our microbiome also undergoes subtle changes. Some diversity goes slightly down. We see a little decline in some of the commensal and beneficial bacteria, and some of the opportunistic pathogenic bacteria may start showing higher abundance. However, research shows that these changes are slightly unique in people that live a longer life. There are centenarians and they live up to 90, 100 plus years of age without any chronic diseases. Their microbiome does not undergo similar changes. They have the ability, or their gut has the tendency to maintain a healthier microbiome during those stages. So there are so many lessons we can learn from this centenarian gut microbiome and how we can implement those into practice, that so we all can foster and nurture a healthy microbiome during adult age, during middle age, stages, so we remain at low risk of chronic diseases. So our lab has proposed this concept. This is a snapshot from a concept paper that we published recently about the bioimaging. We have appreciated the role of inflammaging and immune senescence and senescence as a marker or hallmark of aging, but we believe that even microbiome should be considered as a hallmark of healthy aging, as emerging research has started showing that microbiome plays a strong role in resilience to senescence and chronic diseases associated with aging. So research has shown that, aging comes with a high risk of gut dysbiosis that includes reduced beneficial bacteria, increased pathogenic bacteria, increased chances of leaky gut, causing translocation of bacteria or bacterial toxin from gut to blood, triggering low-grade inflammations, and also that also leads to nutrient malabsorptions that further increases the risk of chronic diseases during old age. However, if this can be prevented by microbiome-based therapeutics through fiber-rich foods, whole-based foods, prudent dietary patterns, rich in polyphenol, probiotics, prebiotics, or unsaturated fatty acids, that can maintain a eubiosis for much longer time, which is important for maintaining the community of beneficial bacteria, which further produces short chain fatty acids, GABA, and other neurotransmitters, fosters healthier nutrient absorptions, and leads to a better gut epithelial permeability, enteric immune system, enteric nervous system, resilience, and that fosters a healthy aging. And that can help us pursue longevity, which is living longer without any risk of chronic diseases. So there have been many research studies that have shown from around different parts of the world that diet always remains a key signature of healthy aging and longevity. There are so many dietary patterns. One of the most successful dietary patterns is Mediterranean dietary pattern, or its related dietary patterns, including Mind diet, Dash diet, uh, Okinawa diet, Nordic diet. All these diets have been associated with hallmarks of healthy aging and longevity. Apart from diet, there are many other factors that we discussed that can shape our microbiome, but diet remains one of the most prominent factors shaping our microbiome, especially during old age. On one side, Western diet patterns have been associated with not so healthy gut microbiome, but vegetarian or Mediterranean diet, because of their richness in specific foods such as whole grains, fruits, nuts, vegetables, as well as legumes, have been associated with healthy gut microbiome associated with aging and longevity. So this is where pulses come into play. Legumes have been part of healthy dietary patterns for ages. But what is not known clearly is, are these benefits mediated via the gut microbiome or not? So that's what we are trying to understand in our lab. For example, this is one of the studies that we did recently, a couple of years ago, where we wanted to see if whether and how these prudent dietary patterns change the microbiome in a good way or not so good way. So we did this pilot study a couple of years ago where we recruited some young adults, middle-aged adults, and we asked them to follow two different dietary patterns, either of standard Western style dietary patterns or a standard Mediterranean diet pattern for just 2 weeks. And we were surprised to see that within 2 weeks, there was a significant increase in the microbiome diversity, in the gut microbiome diversity in the guts, clearly showing that microbiome diet, a prudent diet can really significantly enhance microbiome diversity as early as 2 weeks time. And when we pin down further to identify what is included in this increased microbiome diversity, what are the bacteria that are increased within this increased diversity, we found that there were some specific effects, as you can see on the plot on the right side. There were specific commensal or beneficial bacteria such as Akkermansia, Lactococcus, Leptospira were significantly or insignificantly increased by these dietary patterns, whereas some bacteria that could be potentially opportunistic or pathogenic, such as SMB53 or few other bacteria, were reduced. And there were also some other bacteria that are well-known fiber degraders and short-chain fatty acid-producing bacteria, such as Faecalibacterium, Phoscolarcobacterium, Citrella, and Oscillospora, were found to be increased. So this study clearly showed that yes, Mediterranean kind of diet pattern really boosts the microbiome diversity, and that includes specific nurturing of beneficial and commensal bacteria that are well-known producers of short chain fatty acids and well-known degraders of dietary fiber. We also replicated this study into a more mechanistic model where we did this study in non-human primate models where we had this two group of non-human primates that were maintained on two different diet patterns, for example, Western style diet or a Mediterranean diet style for almost 2 years. I believe this was the only study that has been done in non-human primates for that such a long time. We found that when these non-human primates were maintained on these two different diets, there was again significant boost in the gut microbiome diversity. As you can see on the bar plots in the bottom, bacterial diversity was significantly increased, and within that was increased abundance of some beneficial bacteria. One such example was lactobacillus, which was significantly increased in primates that were consuming a Mediterranean diet, clearly showing that yes, again, there was a beneficial outcome in happening in the gut microbiome because of the Mediterranean diet. And lactobacillus, as we all know, is a very well known regarded bacterium. It is also being used as a probiotic. It's also one of the most widely studied probiotic genera worldwide. But in addition to lactobacillus, we also found many other beneficial bacteria. Their ratios, their population was significantly increased. But not only the microbiome was being benefited by this diet, we also found some systematic benefits. For example, we also performed um, transcriptomics analysis of blood monocytes in these primates, and those analyses revealed that all the genes and pathways related to antiviral immunity were significantly or insignificantly increased when they were eating a Mediterranean diet, whereas inflammatory cascades or mechanisms related to genes and pathways related to inflammatory hallmarks were reduced. And all the genes and pathways that were related to traits, including celiac disease, BMI, body fat percentage, hay fever, allergies, were enriched only in Western genes, but which means that when they were consuming a Mediterranean diet, these traits were reduced. Again, there was a whole body kind of beneficial effect seen. And not only the immunity and microbiome was benefited, we have found that even the brain health was being improved. For example, at the graph at the at the bottom shows that we also found significant improvement in the diagnostic adaptive behavior of these primates. At the baseline, they were all same, but as they started consuming two different diets, the scores started, discriminating very strongly from each other. You can see that all the primates that were consuming Mediterranean diet, their diagnostic adaptive behavior scores was getting improved, whereas in the other group that were consuming Western diet, the scores was very, very low. And what's included in that score is a number of assessments. So this score includes what was their behavior within their tribe, how often they were fighting, the violent experiences, anxiety, depressions, time spent lonely, time spent grooming, sleeping, gait speed. All those behavior assessments were recorded by cameras and observations, and these were all accumulated into one score. So this score clearly shows that the primates that were consuming a healthy diet not only had their healthier microbiome, not only had boosted immunity, but they also were behaving nicely. They were becoming better. They were at a better mental well-being, again indicating some role of improvements in the gut-brain axis and the immune system. So these studies clearly show that why one of the reasons why Mediterranean diet has been associated with healthy aging and longevity might be because it also fosters a healthy microbiome that further leads to better regulation of immune functions as well as the gut-brain axis. And all those Mediterranean diet has been part of human lifestyle for such a long time, and dietary legumes and pulses have been integral part of diet for long. And this is not just new. This has been known for over 2 decades now. For example, once a study was done back in 2004, the Phil study. This study was the first one to show how legumes could be one of the most important dietary predictor of survival in older adults. This study included people from different ethnicities, including Japanese, including Swedish, as well as some other Mediterranean countries, and they categorize all the diets they have been consuming into 9 groups. And you can see that legumes were one of the two significant food groups that were associated with better survival in older adults. And these and the effect of and the association of legumes with healthy aging was significant even when this was controlled for sex, demographic, and other factors. So clearly indicate this was the first study shows that having more legumes in the diet was associated with healthy and longer aging. And this was also replicated in back in 2015 in another NIH survey report that included meta-analysis of 154 dietary surveys from all the places in the world that where people tend to live longer life. And those findings also reproduce the same thing, that most in all those surveys, 95% of centenarians were consuming a plant-forward or plant-based diets that included plenty of beans. So it's been a long time that policies have been associated with healthy aging and longevity. And these are the two. Then this was also reproduced in another study back in 2024, where this huge study from that included people from 9 different ethnicities, including China, France, Germany, Iran, Norway, UK, and USA. It shows that legumes, people that were having more legumes in their diet, they were actually adding years to their life expectancy. As you can see within this rectangular red bar box, um, legumes had more association with life expectancy compared to nuts, fruits, and veggies. So this clearly indicated that how like addition of legumes could add 2, 3, 4, 5 years of life into our lifespan. And most recently, another study, big meta-analysis from over 100,000 people, also placed pulses and legumes among the major 8 food groups that were consistently and significantly associated with healthy aging, intact, better cognitive function associated with aging. These legumes are also associated with better physical well-being, intact mental health, and these were also correlated with lower risk of chronic diseases and increased probability of survival without chronic diseases beyond 70 years of age. So all these studies have clearly shown that legumes have been part of healthy aging for very very long time, and they have been consistently correlated with healthy aging and longevity. And this makes total sense because we know that pulses are prudent food groups. They're of course plant-based, and also they are sustainable because these are affordable, they're accessible, easy to store. They're eco-friendly as well. They're versatile because there are so many ways we can include pulses into our diet, into our culinary and recipes. And also they're a nutrient powerhouse. It's not just the fiber. Of course, they are a rich source of fermentable soluble fibers as well as oligosaccharides, but they're also rich in polyphenols. They also provide those resistant starch, which also fosters healthy gut microbiome. But they also provide a good supply of micronutrients, including magnesium, calcium, iron, as well as vitamins B1 and B3, and also specifically for vegans and vegetarians or people that prefer plant-forward diet. They also provide good source of amino acids. They're also rich in proteins, so their protein digestibility control amino acid scores are also quite comparable to other sources of proteins into our diet. So by virtue of all these, rich nutrient profiles, it can be easily hypothesized that, pulses could also help us recover from dysbiosis or prevent dysbiosis of the microbiome and foster a healthy homeostatic gut microbiome because they're rich in fiber, rich in starches, rich in polyphenols and micronutrients. And all these things are very well known to promote a healthy microbiome. And that's what we are trying to understand in our lab because prebiotic mechanisms, mechanisms of dietary, fiber have been studied for very, very long time. However, the focus has mostly been on fibers coming from chicory roots or other vegetables such as corns, plantains, or sweet potatoes or other sources. However, whether and how dietary pulses can also have prebiotic mechanism, a prebiotic effect on the microbiome remains largely underexplored. And that's what we are trying to do in our lab. For example, this is the first study that we did in our lab where we wanted to study if pulse fiber can also foster a healthy microbiome and can have a prebiotic effect on the gut. So in this preclinical study, what we did was we harvested the gut microbiome of older adults and we transplanted that microbiome into an animal model of aging. So this is an animal model including older mice that were transplanted with human microbiome. And then we maintained these animal models on 5 different dietary lifestyles. One was on a standard Western style diet, CTL stands for controlled diet, without or with incorporation of fiber coming from pinto beans, black-eyed peas, lentils, and chickpeas. And we also had a positive comparison group, which included inulin fiber coming from chicory roots. So we had this six group of animals that were maintained on six different diets. The only difference was the fiber coming from pulses. And after 20 weeks, when we assessed their microbiome, we found that, yes, just like inulin, we found that addition or incorporation of fiber from dietary pulses led to significant or insignificant increase in the microbiome diversity within 20 weeks in this model. Just like we saw in our other studies on Mediterranean diets. Yes, having more fiber from dietary pulses led to increased expansion of microbiome diversity. And within this diversity, it was interesting to see that effects were very specific. And we found that all those four pulse groups were leading to specific fostering of specific beneficial and commensal bacteria and a reduction of potentially pathogenic bacteria. For example, pinto beans, they led to increase of Lachnospiraceae, Immunococcaceae, and Clostridium sensu group while reducing B sulfur Vibrio. Same time, lentil also led to increased expansion of a bacterial family called Lachnospiraceae, which is a well-known human gut commensal known to play a role in fiber degradation and short chain fatty acid productions. Black-eyed peas fiber also led to increased expansion of Lachnospiraceae, Immunococcae, and Oscillospiraceae, all known to digest fiber and promote short chain fatty acids, and also reduce abundance of Enterococcus, which is another potentially pathogenic bacteria. Chickpeas fiber had most profound effect on the microbiome. It led to the increased fostering of many bacteria indicated by green bars, including Bacteroides, Lachnospira, Oscillospira, and many other bacteria known to degrade fiber and produce short and fatty acids. And some other potentially opportunistic pathogenic bacteria were reduced. So again, indicating that yes, fiber coming from pulses did have some kind of prebiotic effect on the microbiome, which means it was increasing the microbiome diversity by increasing the growth of good bacteria and reducing the growth of bad bacteria. But we also wanted to gain an understanding of what's happening at the level of function of these bacteria. And we found that having more fiber coming from dietary pulses actually led to production of more beneficial metabolites, such as short chain fatty acids and neurotransmitter precursors in the gut microbiome. For example, lentils inclusion significantly increased the production of acetate and butyrate in the gut microbiome. In addition, chickpea addition led to increased production of glutamate, which is again a precursor for neurotransmitters. So again, indicated that addition of fiber from pulses was not only fostering a healthy microbiome, but it is also fostering a healthy metabolome coming out of that microbial fiber fermentation. The circular plot on the right side clearly shows the heat map representing all the metabolites that were detected in this study. And this cluster shows that all the four fibers coming from dietary pulses included increased synthesis of choline, glutamate, propionate, butyrate, glutamate, glycine, and other things that are well-known, precursors for short chain fatty acids as well as neurotransmitters in regulating the gut-brain axis. So there are really very positive findings coming out that microbiome, that the pulses from dietary pulses was really fostering a healthy microbiome that was actually leading to a healthy, metabolic outcome from these microbes. But the benefits were seen not only in the gut, but also on the overall systemic health. We also, performed the glucose tolerance test, and we found that there was slight improvement in the glucose levels, in the blood glucose levels in these animals when they were consuming fiber from pulses. We also saw some improvement in the gut barrier permeability after addition of pulse fibers. We also found that behavioral scores such as the working memory, the ability of these animals to remember things, remember previous tasks, or their exploratory behavior to new objects, new experience, exposures, were actually being improved. So there was a better regulation of glucose homeostasis, there was a better regulation of gut barrier permeability, and there was also slight improvement in the behavior health. And we also performed some necropsies on the brain tissues, and we found that the genes related to senescence in the hypothalamus in brain were also downregulated. So indicating that somehow the brain was younger when there was an addition of fiber from pulses into the diet, clearly indicating that how pulses were causing increased expansion of beneficial bacteria and metabolites and neurotransmitters, eventually leading to better cardiometabolic as well as neurological benefits. So these studies indicate clearly that yes, dietary pulses do have prebiotic mechanisms, just like any other fiber coming from plants or fruits or vegetables. Dietary pulses also provide fiber, which leads to beneficial changes in the microbiome and leads to production of short chain fatty acids and neurotransmitters, thereby improving immune health, reducing inflammation, improving gut barrier integrity, um, ultimately, and then also improving senescence, ultimately leading to better hallmarks of healthy aging. But this was a preclinical studies, but this was also, reproduced by some other recently published studies from other groups that also shows that dietary pulses foster good gut bacteria. They promote the growth of beneficial bacteria that produces short chain fatty acids and phenolic metabolites. And these are well known to regulate our satiety as well as hunger. So studies have shown that addition of pulses can also foster satiety and can also suppress food cravings by fostering the regulation of GLP-1 and PYY and ghrelin hormones. At the same time, these compounds produced after this fiber fermentation also improves gut barrier integrity, reduces the risk of low-grade inflammation, and all these things are well known to improve gut health, cardiometabolic health, cognitive health, and foster healthy aging altogether. So propelled by these studies and our own preclinical findings, we are now trying to understand if the same thing will happen in a clinical setting as well. So this is the most recent study that we are doing in our lab, studying the prebiotic effect of pulses on gut and metabolic health in young adults. This is one of the recent studies that we have done in our lab, where we have recruited young adults who are at high risk of dietary fiber gap, which means they're not consuming enough fiber according to the dietary guidelines. And also, we specifically recruited subjects for this study that are not habitual consumers of dietary pulses. So we recruited 60 young adults, and we divided them into three groups. One group was a control group; they didn't consume any pulses during the 12 weeks of interventions. But other two groups were asked to consume 2 to 3 cup of pulses every week, either chickpeas or lentils. So we had total three groups that underwent these dietary interventions with pulses for about 12 weeks. And after that, we measure a lot of assessments to measure their uh, gut microbiome, metabolites, lipid profiles, bile acids, secondary bile acids, as well as oral health. So based on our previous studies as well as studies published by other labs, our hypothesis was that addition of pulses into diet will foster a healthier microbiome, it will increase microbiome diversity, will foster a better gut barrier and gut motility health, will improve lipid profiles and glucose regulations, will also foster the synthesis of secondary bile acid and will reduce, negative or inflammatory cascades. So when we completed the studies, we're still recruiting more subjects. Our analyses are not completed and published yet, but our early research shows some promising effect. For example, we have seen that people that actually included lentils in their group, in their diet, they had a better regulation, some better fostering of beneficial bacteria in their gut. For example, this graph on the left shows that inclusion of lentil into diet increased the abundance of bacteria called bacteroides, Agatobacter, and Bifidobacterium, all three well-known commensal and beneficial bacteria in the human gut within the 12 weeks of time. On the other hand, Prevotella, which could be sometimes opportunistic, potentially pathogenic, was reduced by having pulse into diet. And we found similar effects with addition of chickpeas as well. When subjects that included chickpeas into their diet, they had increased, population of bacteroids, Agatobacter, and also Christianacea and Akkermansia, all very well-known beneficial bacteria in the human gut. At the same time, there also we found reduced abundance of Prevotella, which could be a potentially, which is not supposed to be dominating in the human gut. So again, just like our previous studies, we also found a beneficial outcome because of the pulse additions into the diet. There was a subtle, improvement happening in the microbiome. And when we combine all this data and try to predict like all the bacteria that are well-known producer of short chain fatty acids, such as acetate, propionate, and butyrate, we found that both lentils as well as chickpeas were associated with increased abundance of bacteria that are well-known short chain fatty acids producer. And specifically within this cluster, we found significant increase in the level of bacteria that are well-known producers of propionate. Chickpeas, lentils has much stronger effect than chickpeas, of course, but we found that both groups were causing some increase in the bacteria that are well-known producers of propionates in the gut. And propionate is a well-known short chain fatty acid which has been associated with anti-inflammatory and antioxidant capacities in the literature. So this was again beneficial outcome that we found regarding the effects of dietary policies on microbiome and metabolic output from that fermentation. We also found that there was slight improvement in the whole gut transit time. As I mentioned, we recruited subjects that are at dietary fiber gap, and young adults, especially when they don't consume enough fiber, they also experience, sometimes constipation or uneven gut transit, bowel motility episodes. So therefore, we wanted to see if addition of fiber from pulses is also improving the overall bowel motility or gut transit time. And we found that yes, both lentils and chickpeas actually led to up to 1 to 4 hours decline in the overall transit time per week compared to the control group. So this was a very good outcome that we found that not only the microbiome and metabolites were being benefited, we also found that there was some reduction in the overall gut transit time or bowel motility time with addition of fiber. Some other studies have also shown that fibers from plant-based foods also helps, bowel motility in a good way, and we found the same effect that dietary pulses could also do the same effect here. Previous studies have also shown that addition of fiber also helps with cholesterol assimilation. It also improves lipid profiles. And so we also wanted to see if pulse would have the same effect. And we found that yes, when we measure the blood lipids in these subjects that were consuming lentils or chickpeas, we found that yes, there were some improvements in the HDL, which was highest in lentil groups. So people that included lentil in their diet, they had significantly higher HDL and significantly lower LDL in the blood. So clearly, and this ratio of HDL to LDL and VLDL is a well-known hallmark of lipid profile. So we found that both lentils and chickpeas groups had lowest total cholesterol compared to the control group, and within this, lentils and chickpeas had little higher HDL and lower LDL as well as VLDL. So we found that yes, not only the bowel health was improving or the gut health was improving, these were also, being reflected in the overall cholesterol and lipid profiles in the blood. So early takeaway from our research, ongoing research from all these studies that we're doing right now is that, lentils, of course, was found to have strongest effect on the lipid, profiles. So we found that lentils incorporation into diet led to increased SDL, decreased LDL, so clearly separating from control groups. Although both dietary groups, chickpeas as well as lentils, showed subtle improvements in lipid profiles compared to control groups. So, but we recommend to our clients as well as any other person that this should still be framed as a food and not like a therapy or a dose, because we believe that chickpeas or lentils or any pulses might work best if they're part of a healthy dietary patterns, not just standalone therapy. Number two takeaway that we realized from this study is about improvements in the bowel regularity and fermentation. We found that both pulses tended to show, increase the gut transit time, so they could help with bowel motility. They could help prevent constipation-related issues. Chickpeas had the largest drop in the transit time, up to 4 hours. Lentils had about 1 hour. But within that, we also found that propionate, which is on the prominent short chain fatty acids, was significantly increased with lentils. Although both, pulses led to subtle improvements in short chain fatty acid producing bacteria. And also, we have to appreciate the fact that microbiome is highly, variable from person to person. So just because it worked in one cohort may not work for everyone. So, and we have to understand that this was a otherwise apparently healthy population. So both policies seem to be promoting improvements in abundance of key bacteria, including bifidobacteria, macromencia, Christianelaceae, and we found the reductions in less beneficial bacteria such as Prevotella. But if there is a person that is already suffering from gut dysbiosis, maybe these effects on the microbiome may be even more prominent and profound and more specific to see. So now we are also doing another study where we're trying to bring into practice to provide pulse education, nutrition in children, because just like young adults, just like college students, other age group that is always at high risk of fiber gap are children, especially elementary and middle school children. So right now we're also doing some clinical studies where we are inviting school age children to our lab with their parents, and we are trying to educate them about the importance of nutrition and gut health and the microbiome, and what could be the role of pulses into human nutrition and gut microbiome and overall gut health. So we are conducting this study right now where we are inviting kids, we are cooking pulses with them, we are providing them some ideas for creative recipes. We are cooking with them, and then we are giving them recipe books and handbooks, and they're going home and we are asking them to start consuming pulses, 2, 3 times a week with their families, and we are monitoring and how that incorporation of pulse nutrition into their family lifestyle can change the microbiome, metabolism, overall lipid profiles, gut motility, and everything else. So stay tuned until we complete this study, and I'll be happy to get back and publish and present our findings probably next year. But this is an exciting study because the best way to prevent chronic disease is to start educating about healthy nutrition early on. So we are very excited about this study where we are educating the students, these children, as early as when they are 6 to 7 years of age, and we are trying to incorporate awareness about gut health and nutrition and fiber and pulse into their diet. So our hope is that they're gonna keep sticking to it, and this education gonna be sustainable for the rest of their lifespan, and so it keeps them at low risk of chronic diseases and fiber gap. So key takeaways from all the dietary pulses that we have been doing in our lab are like, pulses do matter because it's not just starch, it's not just fiber, but they're literally whole foods. Um, Dietary fiber always remains a public health concern, especially among young populations that are always looking for convenience, so they always remain at a high risk of fiber gap. So in that way, pulses are highly recognized and appreciated by dietary guidelines as part of any healthy dietary pattern. Now, the recommendation even suggests having half a cup of pulses almost every day, but we recommend to start with like 2 to 3 times per week at least. And it's not just the fiber, they are really nutrient packed with micronutrients, magnesiums, iron, vitamins, polyphenols, soluble as well as insoluble fibers, in addition to resistant starches, as well as, of course, amino acids. And also there is strong and emerging evidence supports that pulses could also, help us meet those fiber gaps. And this addition of fiber into diet is a well-known factor that can help us mitigate the risk of cardiometabolic problems. And it's also a very well-known factor related to fostering a healthy gut microbiome, foster a gut health through fiber and starches. And all these things leads to downstream productions of hundreds of compounds, for example, short chain fatty acids and many other, compounds that are very well known regulators of our intestinal immune functions and our systems, provides us resilience, improves our metabolisms and gut-brain axis, again, leading to healthy aging and longevity. However, we also have to take into consideration age-specific, aspects. Not one shoe we're gonna fit all, so we have to take into account um, appropriate doses for children versus young adults versus older adults, as well as male and females, and for specifically for people that are not habitual consumers of pulses, but now they are interested in adding pulses to the diet. We recommend starting slow, starting at lower doses, for their gut microbiome to adapt. So we recommend, having a quarter cup couple of times a week, and then slowly and slowly every week ramping up a little bit until they reach the guidelines, the dietary guidelines. This helps, gut microbiome and fermentation capacity of the gut microbiome adapt. So it may take sometimes a week or a month for someone's microbiome to adapt to a new inclusion of a new food group into the diet. So because if we start early, we have noticed that sometimes it may lead to some temporary episodes of constipation, flatulence, bloating. So starting slow and slowly ramping up leads to better adaptation of the microbiome and overall gut health, and leading to acceptable doses. We also always encourage into our practice about pairing pulses with some other functional foods. For example, we recommend combining pulses with fermented foods or lean meats or sulfur-rich veggies, vitamin C's and whole grains. It makes recipes more interesting, but it's also science-based because, for example, combining pulses with fermented foods give us both aspects of prebiotics plus probiotics. So pulses provide us fiber and starches, which serve as prebiotic. On the other hand, fermented foods such as kimchi, sauerkraut, yogurt, kefir, miso brings in some probiotic strains. So we get like a whole food completed in terms of pro and prebiotics, and both this together sometimes complement each other by enhancing our gut microbiome diversity, increasing the production of shorts and fatty acids, and overall improving our gut barrier and enteric immune functions. Combining pulses with vitamin C is also a very good science-based fact because vitamin C is very well known to improve the absorption of non-heme iron, and which pulses have. So we always take into account those kind of combinations as well. So combining lentils with bell peppers or citrus fruits or beans with tomatoes or salsa or chickpeas with lemon juice, etc. So this vitamin C reduces ferric iron to ferrous ions, thereby improving overall iron absorption, iron bioavailability, which can help in reducing the risk of iron deficiencies, specifically for people that are vegan or vegetarians and they don't consume enough, heme iron from animal meat products. So this could be a good combination for those kind of clients. Even for those that are habitual meat consumers, pulses could be a good combination with meat because we know about meat factor effects. So combining heme iron with non-heme iron also further improves the absorption of non-heme iron. It also fosters, satiety. So it also prevents overconsumption of meat products. So combining beans with chili, beans in chili with beef or turkey or chicken or lambs or ham, it further improves the absorption of non-heme iron as well as zinc from pulses through the meat factor effects, and overall it together leads to improved mineral absorptions, high protein quality per meal. We get better ability to reduce, total meat intake while maintaining overall nutrient adequacy because both meats and pulses provide complementary vitamins and other micronutrients as well. And combining pulses with whole grains is also science-based. For example, combining black beans with brown rice, lentils with farro, barley, or chickpeas with whole wheat pita. Like we all know that pulses are rich in lysine, but they are limited in methionine. On the other hand, grains, for example, are rich in methionine, but they lack lysine. So this creates a complementary combinations where we check both we check double boxes from the same plate, so it leads to improved overall protein quality and it provides us more adequate dosage of essential and non-essential amino acids from both groups of foods. So overall it also leads to promotion of enhanced satiety as well as glycemic control. So these are a few things that we keep into practice into our lab. And likewise, pulses could also be benefited by combining with sulfur-rich vegetables because sulfur-containing compounds are well known to support glutathione synthesis, whereas pulses, derived amino acids, and polyphenols are well known to support antioxidant and foster a healthy microbial metabolism. So combining lentils with broccoli or Brussels sprouts or onions, garlics, or cabbage or leeks is a good idea because both these things can complement, lead to more nutrient adequacy, which leads to improved endogenous antioxidant capacity. We get more, reduction in oxidative stress and inflammation and support for detoxification as well as cellular resilience. So these are few things that work for our clients that are incorporating pulses into their diet. So that's all from my side for today. With this, thank you again to USA Pulses. Thank you again to Dr. Stokes and Tracy and everyone for inviting me to present my research and talk about pulses and microbiome and gut health. And please visit our lab website and keep looking for publications on these topics in the coming months and years. And thank you to the funders and supporters of our lab. Thank you to my team that conducts a lot of experiments in my lab. My contact information is placed here if someone has any questions and want to reconnect and reach out for any Q&A. I'll be happy to answer any questions. So thank you again. Pleasure to be here. Thank you.
Excellent. Thank you so much, Dr. Nagpal. I was taking so many notes, I've kind of run out of paper here on my desk, and we were flooded with a lot of excellent questions. So anyone still wants to submit a question, you are certainly invited to do that. We will stay on together until about 10 minutes after the hour. We'll try to get through as many questions as possible. Ravi, we had a number of folks who were inquiring about the amount or the dose. And, I was in my notes, you had the unpublished data around the gut transit time was improved with pulses. And then I wrote that you were, I guess, maybe more interested in framing it up as it's not so much about the dose, it's more about the food. We still want to get a target. Is it fair to say a half cup a day? Is that doable? Is that reasonable? Is that going to get us in the ballpark of what you're finding, or what do you think?
Yeah, so in our study, we specifically recruited subjects or young adults who are not habitual consumers of pulses. So
Okay.
we asked them to start very slow. Uh,
Yes.
we started with quarter cup per week for the first week.
Okay.
And then second week was half a cup per week. And then 1 cup and then 1.5 cup. So by fifth week, then they were within the dietary guidelines of
Mhm.
2 to 3 cups per week. And
Got it.
this increased the adherence and acceptability among these participants. We didn't want to start stress. So we gave them 4 weeks of acclimatizations. And by fifth week, they actually reached the actual recommendation that we make, like 2 to 3 cups if they are males, or 1 to 2 cups per week if they are females, or 1.5 to 2 cups. So, but we waited, we gave them 4 week of acclimatization and ramping up phase, where they were given freedom to start like either quarter a cup or half a cup just per week, just to get acclimatized. And then second week slightly higher, third week slightly higher, fourth week slightly higher. But by fifth week, they were actually into our dietary guidelines, recommendations.
Okay.
So this worked for them, and this also led to increased adherence. They were happy following our recipes, because in the past we have experienced that if you start all fresh, like if you, all of a sudden you start adding a new food group, especially rich in fiber into your diet, sometimes people can experience temporary bloating or flatulence or other indigestion or intolerance, and they don't want to consume it anymore. That leads to a negative, feedback and thought process. So we wanted to prevent it. So we started very slow. We gave them 4 weeks of adjustment, and then they started following the dietary guidelines.
Thank you. Thank you for that. Robbie, we have someone here who asked early on. She mentions there's a suggestion that fiber could be considered an essential nutrient because of its essential role in the microbiome. Do you have an opinion about that?
I agree with that to some extent, that the fiber is an essential nutrient, but let's appreciate that it's not just about the microbiome. Fiber has a number of many other benefits as well. It's not only it fosters a healthier microbiome in the lower gut specifically, and that fermentation leads to production of short chain fatty acids and neurotransmitters. But even without fostering the microbiome, fiber also helps us, maintain our bowel movement.
Mhm.
Gut. It also helps in cholesterol assimilations and bile acid regulation. So inclusion of fiber does have a number of benefits in the overall beneficial benefits in the gut health as well as metabolic health. So I totally agree that yeah, fiber is indispensable to overall gut health and bowel health, and I think it is essential nutrient, and that's the reason we are seeing more emphasis in the dietary guidelines. Also, this time that there is more emphasis on plant-based and fiber-rich foods, not only for gut health but also the overall health.
Thank you. I
But
got a sense, I
but I would
scanned the
also
list. Oh, I'm
yeah,
sorry, go ahead.
I would also like to add that it's not just one fiber. We need to have a variety of fiber,
Yes.
ideally, to our diet to foster a variety of bacteria in the gut. So a little bit of fiber ideally should come from plants, a little bit from fruits, salads, and pulses, chickpeas, etc.
Mhm.
So because every source provides us a different chemistry of fiber, some fiber is galacto-oligosaccharides, fructo-oligosaccharides, resistant starch, complex carbs, soluble carbs. We need a variety of fiber also. So therefore, it's very good idea to keep on switching between different, choices of fruits, vegetables, salads, chickpeas, lentils. That gives a variety of fiber, and that maintains a huge diversity of the microbiome. Because just like us, our microbes also have their own favorite and least favorite foods. So one fiber may not be able to promote all the beneficial bacteria in the gut. So we need a variety of fiber as well.
Thank you, Robbie. That message was also noted in the imagery and in the foods and that you talked about toward the end of the presentation. We could clearly see a tremendous amount of variety. I was going to also mention that I scanned the list of people on today. It's tons of dietitians, and I think they're all going to be fans of fiber. And I think they'll all support you in this campaign to make fiber an essential nutrient. So thank you for that. We have another question here from someone who says, you mentioned, Ravi, the difference in commensal bacteria in babies born by cesarean delivery. Given that every gut microbiome is unique, are the differences clinically relevant in the long run?
So in our study, we found that yes, babies that were born by C-section, they lacked a number of beneficial bacteria that we typically get colonized with when we are born vaginally. So we require a lot of, for example, lactobacillus. Most of the lactobacillus in a newborn baby comes from maternal vaginal microbiome because this vaginal microbiome is rich in lactobacillus, and this lactobacillus becomes a fundamental member of our gut microbiome for the rest of the lifespan. So we found that kids that are born by C-section or preterm, they lacked some of those lactobacilli. Rather, they had more of maternal skin bacteria, including streptococcus or staphylococcus in their gut, because C-section leads to more exposure to contamination with skin microflora. Um, we didn't, so we followed up these babies for up to 3 years. Well, unfortunately, 3 years is not a long enough time for us to detect like any clinical outcome because we don't do clinicians also don't perform a lot of diagnostic tests during 3 years. But we did found that the kids that were born by C-section, they had slightly higher weight by the time they turned 3 years of age compared to kids that are born by regular full-term delivery. So that was one indicator that we found. Probably maybe in the long term, I don't know. We could not follow beyond 3 years, but it might be that some other studies have also shown that babies are born by C-section, and then after that, they're also not exposed to exclusive breastfeeding, which also fosters healthy microbiome. They're usually at higher risk of developing obesity. They're also at slightly higher risk of developing atopic or allergic conditions because they lack some bacteria that trains our immune system during first 1 to 3 years of development. So it's not just our studies. There are also a number of other studies that have been published that shows that preterm birth as well as C-section birth leads to some, missing of some lack of beneficial bacteria that also disturbs the microbiome and immune crosstalk, and that may increase the predispositions to development of allergy, atopic conditions, or adiposity in later years. But during those later years, many other confounding factors may also come into picture. For example, studies have also shown, and even in our study, we found that kids that were born by C-section, but then they were exclusively breastfed for 3 to 6 months of their life, first, month, they were better off in terms of their microbiome. They still had healthier microbiome compared to kids that were born by C-section, but then were also exposed to formula feed. For some reason, they could not be exposed to breast milk. So there are
Okay.
also following up factors, the foods that the baby eats during first few months, and the weaning foods and environment, the hygiene status, siblings, pets, those things may also come into picture, as well as any exposure to medications or antibiotics may further disturb that predispositions.
Thank you for that, Robbie. And we have time for one last question. Is the age-related decline in gut microbiome diversity, is that due to reduced fiber intake?
So reduced fiber intake is one reason, but there are many other reasons because as we tend to grow old, our bowel, our stomach pH changes, our appetite changes, domestication also changes, poor
Mhm.
oral health can also lead to less mastication and chewing, and salivary enzymes change. So the kind of foods we can, not only the kind of food that we can ingest or metabolize change, but also the way we metabolize foods changes. So not only in so microbiome is an outcome of many things changing during the old age. So mastications, then we also fall prone to more medications. Polypharmacy also leads to some decline of some bacterial communities in the gut. That also leads to intolerance to some of the foods that we used to be able to digest and metabolize normally when we are young. But now, since some of the bacteria are low in proportion, we cannot digest a lot of fiber-rich foods. We don't feel hungry. Uh, so it's a number of factors related to aging, lifestyle factors, diet factors, mastication, stomach changes, proneness to sickness, and then proneness to polypharmacy. All these factors together lead to changes in the microbiome, and that further leads to lack of, tolerance of fiber-rich foods.
Got it. So it's fiber and yes, and.
Yeah.
Okay. Thank you. Well, with that, I'm going to wrap us up for today. I just want to say thanks once again to you, Ravi, for the presentation. Excellent content, excellent material. Also like to thank our audience for joining us and for your engagement, submitting questions. This concludes today's webinar. Have a great day, everybody. Goodbye.
Thank you.